Okay. Hello everybody. Sorry for the late start. I'm Chao-Po from ShanghaiTech University. It's my great pleasure to introduce our first speaker, Dr. Yang.
RNA collaborative to me. You can turn your camera on for the intro.
Okay. Okay. So Dr. Yang is the professor at East China University of Science and Technology and the deputy director of the State Key Laboratory of Bioreactor Engineering. He graduated from Tsinghua University and did his postdoc training at Harvard Medical School. And he joined the faculty of School of Pharmacy, ECUST in 2006. His research is focused on synthetic biology and chemical biology methodologies for fluorescence imaging and optogenetical manipulation of living cells, and their applications including anti-cancer drug screening and biomanufacturing. So his title will be Imaging RNA Dynamics with Bright and Stable Fluorescent RNAs. So let's welcome Dr. Yang.
Thank you very much, Chao-Po.
Hi, so you can start.
Okay. Good evening, everyone in China, and good morning, everyone in United States. Thank you, Chao-Po, for the nice introduction and the invitation to the RNA Collaborative Seminar. It is my great pleasure to share our recent works on fluorescent RNAs.
As we all want to share the screen, we haven't seen your slides yet.
Oh, I am sharing it. Let me see.
Oh.
Can you see it?
Yeah, it's good.
Oh, nice. So let me continue. As we all know, fluorescent proteins has been essential not only for the analysis of protein functions and dynamics, but also for the detection of ions metabolites and protein interactions in living cells and in vivo. Since their first development in 1994, fluorescent proteins have been widely used and revolutionized the living cell and living organism imaging. In 2008, fluorescent protein imaging methods was awarded Nobel Prize. Similar to proteins, RNAs can fold after transcription and undergoes many events such as splicing, editing, modification, degradation. They also interact with proteins and small molecules. Thus, RNAs are highly complex distributions, behaviors, and functions in cells. A robust fluorescent protein-like approach for tagging RNAs to monitor RNA spatial temporal dynamics in living cells would be especially useful for understanding their functions and mechanisms underlying diverse biological processes. So, fluorescent protein was found in jellyfish, coral, or even eels. However, unfortunately, the RNA equivalent of fluorescent proteins was not found in nature. When we studied the classic fluorescent protein from jellyfish, we can observe that layer fluorophore are formed by the oxidation of peptide chains, and the fluorophores such as formed embedded in these beta barriers of fluorescent protein, and the colors of fluorescent proteins are determined by the structure of the fluorophores. 10 years ago, Miyawaki's group discovered a new fluorescent protein named UNAC. This protein was isolated from eel. The UNAC binds with bilirubin, a natural fluorophore found in our bodies. Interestingly, the fluorophore in both these two classes of fluorescent proteins only fluoresce when they are tightly bound by proteins. Otherwise, this fluorophore shows no fluorescence when they are alone in solution. So we can conclude that the fluorescent proteins are made of fluorogenic dyes and proteins tightly bound with them. So this inspires scientists to search for RNAs that can specifically bind with fluorogenic dye and to develop fluorescent RNA, the RNA equivalent of fluorescent proteins. Early work started more than 20 years ago. For instance, Roger Qiang studied an RNA aptamer with nucleic acid intercalating dye named magenta green. Binding with the RNA aptamer to the fluorophore leads to thousand-fold of fluorescence enhancement. While such a concept has been recognized since then, many of nucleic acid binding dyes or the fluorophore quencher conjugates have shown low turn-on ratio and significant background fluorescent leaf cells. And sometimes these dyes can hardly diffuse across plasma membrane. So Simon Jeffrey in Cornell and co-workers pioneer in RNA mimics of GAPs. These are aptamers that specifically bind to non-fluorescent analogs of GAP fluorophore and activates its fluorescence. So this fluorophore can diffuse easily into the cells and fluoresce when the aptamer was expressed in these cells. However, their fluorescence appeared to be dim and works only for high abundant RNAs. We reason that many of these challenges for development of robust fluorescent RNA may originate from their low binding affinity and the thermal stability when they express in cells. As it is difficult to lock small and flexible GAP-like fluorophore firmly in the overall structure of the RNA aptamer complex. We therefore designed this HBC dye with a structurally rigid electron donor and a strong electron acceptor. So this HBC is non-fluorescent in solution, but it became strongest fluorescence upon constraining intermolecular motion. For example, when we add glycerol to increase the viscosity of the solution, the fluorescence of HBC dye is significantly enhanced. So we then proceed to select for aptamers bound with HBC using the SELECT approach. Finally, we identify an aptamer that it can enhance HBC fluorescence more than 3,000-fold. So we then minimize and optimize this such aptamer and call it PAPR. So PAPR is a small aptamer with a core structure only 40 nucleotides. It shows fluorescence color from cyan to red when it bound with HBC dye analogs. These analogs were made by tuning the aromatic pi structure and adjusting the electron donor and acceptor capacity. So mammalian cells expressing PAPR exhibit robust fluorescence on incubation with HBC dyes. So our collaborator, Dr. Aimin Ren in Zhejiang University, solved the crystal structure of PAPR. As expected, the near planar fluorophore molecule intercalates in the middle of the structure of the PAPR RNA. So this explains its bright fluorescence. Cells expressing PAPR showed 10-fold more enhanced fluorescence compared to broccoli or corn, the best fluorescent RNA at that time. So we then made PAPR in tandem arrays, which enhanced fluorescence signal another tenfold. With these combined efforts, brightness of cells expression PAPR fluorescent RNA showed more than 100-fold enhanced fluorescence. So we then fuse pepper to scaffold RNA, such as tRNA or F30 or other functional RNAs, and found that such fusion showed minimal perturbation of RNA localizations. For instance, ribosomal RNA and mRNAs are localized in cytosol, while small nuclear RNAs localized to nuclei. When we test the fluorescent RNA and fluorescent protein together to the various subcellular compartments, we found that they showed nearly identical localization. So this data showed that we can label and image RNAs just as simple as proteins in live cells. We reported the Cleaver, a bright, long stop-shaped fluorescent RNA last year. Cleaver is rather small. Its core structure is only 28 nucleotides. It's bound with NBASI fluorophore and showed orange to red fluorescence. Cleaver showed bright fluorescence in these cells as its folding was not mechanism dependent. Again, the crystal structure showed that NBASI dye is tightly bound by the Cleaver RNA. Cleaver shares excitation spectra similar to those menin peppers, but have large stock shift emissions, sometimes is exceeding 100 nanometer. So this enables them to carry out a single excitation dual color imaging of cleavers and peppers using a single photon or two photon microscopy. So by this means, we can minimize the phototoxicity of dual color imaging. Most cases, you need two lasers, but here you need only one laser due to the long stop sheet properties of a Cleaver fluorescent RNA. So here we show that the two mRNAs and the two proteins can be imaged together by full color imaging in a single cell. We found that Cleaver was perfect for label small nuclear RNAs, such as 7-SK, U6, or U2, due to its very small size. So image showed that these RNAs localized in nuclei and beautifully co-localized with their binding proteins, such as SC35, STAR3, or colin. We then use Cleaver U2, U1 to monitor U-body induction. The lead cell kinetics which have not been reported before. So this data, we can observe the appearance of these U1 U-bodies when the cell is under stress, and we can observe that this fusion of the different granules. So this data supported the hypothesis that these U-bodies represent liquid phase separation, and they can fuse. When we remove the stress, these U-bodies would disappear. Cleaver have ideal spectral characteristics to serve as a bioluminescence resonance energy transfer acceptor for nanolook. So its excitation overlaps with nanolook's emission. So if a Cleaver RNA and a nanolook protein are in close proximity, the excited state of the oxidized reaction immediately of the nanolook active site would be transferred to excite Cleaver by this bright phenomena. So which provide a novel method for real-time detection of RNA protein interactions. So we engineered the MCP protein to optimize the breadth between a nanolook MCT fusion protein and MS2 Cleaver fusion. So we can observe this spread efficiency is rather high. So we use this system. We can observe the RNA dynamics in a live animal. So this time is a direct real-time RNA imaging in live animal, which cannot be performed by other techniques. So this spread phenomenon between nanonucleus and cleaver can also be applied to dissect RNA protein structure and conformation. So we studied here the U1 RNA interaction with the U170Ku1 protein. We observed that this when we fuse the nanonucleus to the protein, which is close bind to the stand-up one, we observe that this stand-up one interacts with this stand-up 3, but not stand-up 2 or stand-up 4. This can be done by in vitro study or in live cell imaging. So we all know the most widely used fluorescent protein is the green fluorescent protein. So it's widely studied and widely used in biochemistry, cell biology. Current green fluorescent proteins include spinach, broccoli, and also the pepper 530 we developed hormone. The pepper is the most bright, the brightest fluorescence. However, this the wide applications of this fluorescent RNA has been hindered by limitations such as fast photo bleaching or photo switching. There's an inadequate cellular brightness and dependence on the magnesium, because we found that this broccoli fluorescent RNA and the pepper fluorescent RNA, they need magnesium to enhance their fluorescence, but the magnesium is limited in a live cell. So we now report, just last month, we report a new green fluorescent RNA occur, which was selected by a small molecular fluorophore ACE. This is one of the smallest fluorophore. So occur showed bright green fluorescence with emission of around 505. It's bind with ACE with very high affinity. The KD, the constant of dissociation is 1 nanomolar. It's extremely low, and this occur is thermostable than broccoli and pepper, and it is also magnesium independent, unlike broccoli or pepper. So when this occur, it was expressed in live cells, it exhibits 10-fold or up to 30-fold higher fluorescence than the other two fluorescent RNA. So we found that for the pepper fluorescence is about 1/3 of is about, let me see, is 1/7 of the occur. But when we found that when we incubate these cells with magnesium, the pepper fluorescence can significantly enhanced, equal to the fluorescence of occur fluorescent RNA. When we tag to the mRNA, the occur fluorescence is bright, and its fluorescence further increased with more tandem copies of occur was tagged to the mRNA. We can observe nicely the colocalization of occur fluorescence and the protein, the amenteric protein. This mRNA is present in the same cells. We found this occur is actually about orthogonal to pepper 620. So occur doesn't bind with the HBC 620, and the pepper doesn't bind with the AC dye. In deep cell studies, we can observe, we can perform dual color imaging of RNAs with actually no interference or leaky signals from the other fluorophore. Further, we found the ochre showed much enhanced photo stability. More than 90% of fluorescence was preserved after continued illumination, while the fluorescence of other fluorescent RNA dropped to minimal. So for this reason, ARC allowed structured illumination super resolution imaging because it's very photo stable. Examination of the stress granules with this structured illumination microscopy revealed that both these proteins and RNAs, they exhibit non-uniform distribution in stress granules with clusters of higher concentration. So this is actually the first time to study the detailed the structure of a stress granule using RNA imaging. So this is the table for the characteristics of green fluorescence RNA available today. Indeed, the aqua is superior in brightness and also the very high affinity to its ligands. So at date, they are hundreds of fluorescent RNA proteins available. So they have different colors, infrared for the in vivo imaging, brighter, stable fluorescent proteins are still highly desired. Some fluorescent protein has unique properties such as photo conversion or photo switching. So I believe the fluorescent RNAs development will follow the fluorescent proteins lineage pathway. In addition to multiplex and single molecule imaging of RNAs, this fluorescent RNA can also be developed as sensors just like these fluorescent proteins. So here we show that by harnessing CRISPR's capacity, these fluorescent RNAs can be used for genome loss imaging. So here we label two sites. We use the pepper and the cleaver for dual color imaging of this genome loss. We also engineered a series of metabolite sensors based on the pepper and the ribose switches that combines with this small molecule metabolites. So we just fuse this pepper and with this ribose switches, the binding of the metabolites will induce the formation of purple pepper conformation and stimulate the fluorescence of pepper. So these sensors showed up to 100-fold of fluorescence enhancement when binds to these metabolites. As an example, we measured an important methyl group donor important for epigenetics. So our sensor was bright in cells. When the SAM synthesis was inhibited by a chemical, this fluorescence drops immediately. When this inhibitor was washed out of the medium, the fluorescence recovered. So this data showed that this metabolite sensor based on fluorescence RNAs is useful for for the study of metabolism. So here is another interesting image video we just acquired. So the green signal is the ER is a protein localized in endoplasmic membrane, and these pink dots is single molecular RNAs. We can observe that these RNAs indeed a part of them are localized to the ER membrane, and a part of them diffuse freely. So according to this data, we can examine the translational status or the ribosome binding status of these mRNAs. So in my lab, we aim to develop all optical approaches to monitor and control RNA biology. We also develop light optogenetic tools that we can control the transcription, degradation, translation, splicing of RNAs. So currently, we can use light to control every step of RNA metabolism. So we want to so as these fluorescent RNAs and the optogenetic tools are all encoded by genes, they can be easily introduced to leaf cells and used in vivo or even in transgenic animals. So at last, I would thank my team members. Dr. Liang Zhou is highly advanced, expertise in the fluorophore design. So and Dr. Chen Xianjun, Da Shen, Suni, Jiang Li, Xixin, Dr. Zhou Fangting, they all participate in this RNA fluorescent RNA studies. I would also like to thank my collaborators, Dr. Ran Aimin, Huang Xinyi, Song Qian in Zhejiang University, and also professors Tang Yun, Do Wenli in my university, and also my mentor, Joseph Noscalzo in Harvard Medical School. I also like to thank my grant that support me, this grant from the National Natural Science Foundation of China, Ministry of Science and Technology, and Shanghai Science and Technology Commission. So, and last, I would like to thank you for your attention.
Okay. Thanks, Dr. Yang, for this very novel work. Due to the time limit, I will just pronounce one of the Q&A, and I think there are several ones, and I think Dr. Yang will be very happy to answer it afterward. So the first question is, what's the current lowest abundant RNA you can confidently visualize in live cells using Aqua?
So actually, Aqua has the single molecular imaging capacity. So even there's one molecule of RNA, you can image it in cells. So wow, single molecular imaging. Yeah,
that's very impressive. Okay. So thanks, Dr. Yang. We started late, so we have to switch to Dr. Liu. Thanks for your talk. So please answer the question, type the answers in the Q&A section.
Okay.
So, okay. So okay, you can stop the screen sharing, and we will switch to Dr. Liu. Okay. Dr. Liu, can you? Oh, okay. Okay. You can share your screen. Okay. Okay. We still see those black boxes, so you may have to move it a little bit. Okay. So during that time, we'll do some introduction. So Dr. Liu received the PhD degrees from Institute of Biophysics, Chinese Academy of Sciences. He completed his postdoctoral training at Washington University School of Medicine and continued his research at Harvard Hughes Medical Institute and the University of Pennsylvania School of Medicine as a research associate. Dr. Liu joined the faculty of School of Life Sciences, University of Science and Technology of China in 2012. his laboratory is interested in elucidating the physiological mechanisms underlying pathogenic mechanisms of neurodegenerative disorders, such as Alzheimer's disease. So that's also his title, The Role of Non-Coding RNAs in Brain Aging and Alzheimer's Disease. So, Dr. we still see those boxes. We will see if it affects to that much. There's the one in the right down, there's a big box over there.
Okay.
So can you...
Sorry, yeah, sorry about that. Maybe
okay,
we
so anyway, that's just the start. Let's see. Hopefully it doesn't bother that much.
Yeah, hopefully,
okay.
Yeah, won't
Okay,
bother you
thank
much.
you. You can start.
Okay. Thank you for your introduction. This is John from USTC. So I'm going very briefly share with you my lab's recent work. So my lab is really focusing on the role of non-coding RNA in brain aging and neurodegenerative disease, especially Alzheimer's disease. So why are we doing the brain aging and the related disease? So the whole world is getting aged. So especially in China, the aging population increased dramatically. The left panel is the projection from 1960 to next 100 years, how the aging population increased dramatically, especially in China. So the whole generation is actually getting old. And also relate to the aging, many aging-related disease, especially brain-related aging disease like neurodegenerative disease, Alzheimer's disease. So in the world, actually, especially in America and in China, the aging population related to Alzheimer's disease is increasing dramatically. And also some recent studies show for Alzheimer's disease, the diagnosis is getting younger. It seems like before this decade. So for AD, age for AD is around 65 years old, but now it's getting younger, like 55. So the reason is not clear. So why we study this and how to study this, especially the non-coding RNA, AD, and brain aging. So the first study we do is about ApoE. So for Alzheimer's disease, there's two types of AD. One is called early onset AD. We also call familial AD. So onset age is about 40 to 60, mostly related to APP and presenilin mutation, but only 3% of patients belong to this group. About 97% patient is actually belong to another group. It's called late onset AD. So the onset age is about...
sorry, Dr. Liu. We just have a black box on the right down side. I saw you when we prepared that you remove it before. So if you can just make it smaller, not on the one on the top is okay, but the one at the right down, there's a big box there. So if you can just move it the right word or something, then that's going to be helpful.
Right. I'm not sure. Yeah, I just cannot see that box.
Okay.
So that's
okay. Okay. I can share a screenshot to you.
okay.
If we cannot get over that, yeah, it happened when we prepared the slides. Let's give it a try. So do you see where it is?
Right. Okay.
Uh,
Yeah.
okay.
I'm not sure what's going on. Yeah, because in my screen, it's nothing there.
There's nothing there. Okay. Okay. So if you
Yeah.
cannot fix it, that's just keep going. With
Okay.
something.
Yeah. Right. Yeah.
Okay.
Maybe we
Sorry.
just.
Sorry.
It's
Sorry
okay.
for.
Yeah. Maybe just keep this way. Yeah. Let's just. Okay. Uh. Okay. We are basically working on the ApoE for many years. So the major function of ApoE is a lipid carrier. The major function of our ApoE is carry lipid from astrocyte to neurons. So on the neuron, the receptor for ApoE we call ApoE receptor, like LRP and the LDL receptor. They can uptake ApoE particle and internalize and deliver to lysosome and degrade them, and also release the lipids. So the neuron can use lipids for the membrane structure. So the major question in the field is we know ApoE, especially ApoE4, is a major risk factor of Alzheimer's disease, but we don't know why. Why ApoE4 or why ApoE? How ApoE regulates the pathology in AD? So to solve this question, to answer this question, we basically did some research, especially we can culture astrocyte in vitro and purify ApoE particle from the conditioned media and use this ApoE particle to treat neurons, see what happened to neuron. Then basically we did RNA-seq after ApoE treatment. So after RNA-seq, we found RNA expression level is changed dramatically, especially many gene expression downregulated. From the downregulated genes, we did a Go analysis. We found many genes related to lipid metabolism. So red panel is the cholesterol biosynthesis pathway. So it's from substrate acetyl-CoA through like 20 different steps, then become a cholesterol. So many enzymes in this pathway decreased after ApoE particle treatment, means this pathway is silenced. Then the substrate acetyl-CoA accumulate. So the final result is acetyl-CoA transfer to nucleus and promote histone acetylation. So basically, we found that ApoE particle can promote neuronal histone acetylation. Why is that? Then we analyze ApoE particle. We found ApoE particle contain many different things, especially ApoE protein and many different lipids. So we use this protein and the different lipids to treat neuron. Found no gene expression change. Then there's something else in this particle to regulate the gene expression. Then we found this component is RNA species, especially microRNA. Then we found astrocyte actually can use ApoE as a vector to carry microRNA from astrocyte delivered to neuron to regulate the neuronal expression. So we analyzed like what kind of microRNA inside of an ApoE particle. We did RNA-seq. We found many microRNA especially in the ApoE particle, like 100 different microRNAs. Then we use the target analysis method. We found different microRNA has different targets, but many different microRNA can target similar targets, like the enzyme in this pathway. For example, in this cholesterol biosynthesis pathway, HMG-CR is the key enzyme, rate-limiting enzyme. This enzyme can be targeted by 13 different microRNA in the ApoE particle. Means ApoE particle can carry many different microRNA to silence the pathway. So we already know the substrate acetyl-CoA accumulate and promote histone acetylation. The next question is what kind of gene histone acetylation is increased? Then we did a chip-seq. We found many learning memory related gene, the acetylation level is increased. So we can just basically summarize here. The astral side can release ApoE particle. This particle can carry microRNA, get into the neuron to silence this pathway and promote histone acetylation to regulate the gene expression. So another question is, we know human has different ApoE isoform, like E2, E3, and E4. Different ApoE isoform has different effects or functions in the AD pathology, especially ApoE4 is a major risk factor. Then we can purify different ApoE particle and use a different ApoE particle to treat neurons, see what happens. Then we found ApoE3 particle has a stronger effect than ApoE4 in terms of the lipid metabolism regulation and histone acetylation. Then we use EM to analyze different ApoE particle and compare E3 and E4. We found E3 particle is actually much bigger than E4, meaning E3 contains many more microRNA than ApoE3 can have a stronger regulatory effect than ApoE4 in terms of histone acetylation and gene expression regulation. So this the first part of work about ApoE particle can carry microRNA to regulate neuronal function involved in Alzheimer's disease. So the second example I'm going to briefly talk about is about the lincRNA, long non-coding RNA. So the initial interest is about how and what kind of lincRNA can be involved in brain aging or Alzheimer's disease pathology. So during brain evolution, we found lincRNA is expanded in the brain dramatically, especially in terms of quantity and quantity. So the first lincRNA we are identify and categorize is LONA. So the name is from these 4 English words. So we use this simplified name called LONA to describe this long non-coding RNA, because this particular lincRNA is only solely localized in the nucleoli. So that's why we name this lincRNA as LONA. So the initial function categorization, we found LONA actually can regulate the synapse plasticity and also involved in the learning memory regulation. Then has some role in AD pathology, but we really don't know how LONA can regulate this gene expression, especially in nucleolars, and what's the molecular mechanism for LONA for that to work. So the study we start is from the cell biology. Then the first thing we do is we can knock out LONA in cell. When we knock out the LONA, we found that nucleolar structure is actually disrupted. We know nucleolar structure has 3 layers. The inner layer is called FC. The middle layer called DFC, the outer layer called GC. So NPM1 is the outer layer marker. When we knock out LONA, NPM1 is actually released from the nucleus to the nuclear plasm, meaning the nucleolar structure is actually disrupted. Then we did some in vitro study to see why and how LONA involved in the nuclear structure, especially nucleolar assembly. We found LONA actually interact with the 2 proteins, one called NPM1, is another one is FBL, then can promote the nucleolar assembly. When we knock out LONA, NPM1 released from nucleolar into the nuclear plasm. The NPM1 is actually soon get acetylated. We call acetylated NPM1. Acetylated NPM1 can bind to the chromatin and recruit another complex we call PRC2 complex. Then PRC2, the major function of PRC2 complex is the methylate H3K27. Then we found after this particular histone acetylate methylation, the gene expression is actually done. So we can use this cartoon to briefly go through this process. So LANA in the nucleolar is particular in the middle of DFC and GC. So this particular localization has a very unique function. When we knock out the LANA, they can disrupt nucleolar assembly. Then NPM1 released from nucleolus, then very soon get activated by P300. So activated NPM1 can bind to the DNA and recruit a PRC2 complex. Then methylate H3K27, then downregulating expression. So basically, this study answers the question about LANA, like how LANA regulate gene expression in nucleolar. Finally, I'm going to spend a few minutes to talk about another work about the tRNA fragment. So we know tRNA is really critical for protein translation, but many recent studies show tRNA is actually can be cleaved by many different enzymes to become tRNA fragments. So these fragments actually accumulate in brain aging and also in many different diseases. These tRNA-derived fragments can play a very important role in the pathology, but how, especially at the molecular level, like how these tRNA-derived fragments can regulate or involve in pathology is not clear. So this study is actually set up to answer this question. The first thing we do is we take a aging brain and compare to the young brain to see what kind of tRNA fragment can be accumulated in this aging process. So after sequencing, we found one particular tRNA fragment is accumulated very dramatically. It's called glutamate-derived tRNA fragment, meaning glutamate tRNA is got cleaved and produce this fragment and accumulate in the brain aging process. So the right panel is the northern. We found in the different age, especially 22 months old mouse brain, the glutamate tRNA-derived fragment accumulated dramatically compared to 3 months old mice. Then the first question we want to answer is how is the tRNA fragment and why is this particular tRNA fragment is coming from? Through the screening, we found the ANG is enzyme is responsible for this tRNA cleavage. We found ANG is actually phosphorylated in the young brain. When the brain getting older, the phosphorylation is lost. Then ANG transferred from the nucleus to the cytoplasm. Then the cytosolic ANG can cleave glutamate tRNA and produce glutamate tRNA fragment. So this particular glutamate tRNA fragment is actually in the cytosol. So we use another method called FISH to visualize where this particular tRNA fragment localized in the cytosol. So to our surprise, we found this glutamate tRNA fragment is actually mostly in the mitochondria. This co-localization with a mitochondrial marker called TOM20, and also this particular fragment. Then the question is, how this particular tRNA fragment transferred to the mitochondria. Then we did a pull down using Anderson's RNA to pull down this particular tRNA fragment. Then we did a mass spec, see what kind of protein associated with the tRNA fragment. We found the major protein associated with the tRNA fragment is called LARS2. The LARS2 is actually the enzyme residing in the mitochondria. The major function is to put leucine to the tRNA. So it's inside of a mitochondria. Then the tRNA fragment can compete with LARS2. Then the LARS2 is kind of a loss of its initial function. Then this final result is the mitochondrial translation is disrupted. Why is LARS2 is so important for mitochondrial translation? So we analyzed all 13 proteins can translate in a mitochondria. We found a very common feature for this 13 mitochondrial protein is the leucine content is really high. The number one is ND3. It contains about 25% leucine residues. That means if you disrupt the leucine amino acylation, then it affects mitochondrial translation. And also, we did we used a Western blot to analyze all 13 different mitochondrial proteins. We found they all decreased after this particular tRNA fragment accumulation. So the final question is, what happened to the mitochondria after this particular tRNA fragment accumulation? We found normally mitochondrial structure is like this shape structure, but after this particular tRNA fragment accumulated, the crystal structure, the inner memory structure called crystal. The crystal structure is actually much bigger, like a bubble. It's got disrupted. Then the final question is, we know this particular tRNA fragment produced in the cytosol transfer to mitochondria, affect the mitochondrial translation. Then what's the relationship about this glutamate tRNA fragment with the glutamate level, because it's a glutamate tRNA. So to our surprise, we found an enzyme called GLS is actually a glutamate biosynthesis enzyme. This particular enzyme is actually residing on the mitochondrial inner memory, also called crystal. When the crystal structure got disrupted, GLS can be released from the crystal and very soon got degraded. Then we understand if the enzyme, especially biosynthesis enzyme, is getting degraded, the glutamate level, glutamate metabolism is affected. Glutamate level is actually decreased. So we also analyzed the glutamate level in the mouse brain. We also found the glutamate level is actually decreased. And also this EM structure shows glutamate containing vesicles. This glutamate containing vesicles also decreased. So we know glutamate is a major neurotransmitter for the brain function. If the glutamate level decrease, it's definitely affect brain function. Then the major function we are studying is the learning memory. So we analyze the learning memory behavior in this old mice or aging mice. We know aging mice has a learning memory impairment, and also this glutamate-derived fragment is actually accumulated. If we target this accumulated glutamate tRNA fragment, can we rescue the learning memory impairment? The answer is yes. So we designed the ESO can target this tRNA fragment and put it into the brain. Then we found not only rescue learning memory impairment, also can rescue the mitochondrial structure. The Christie structure becomes more normal. So finally, just spend a few minutes to summarize this part of work. So during brain aging, we found a key enzyme called ANG. Normally, it's phosphorylated inside of nucleus, but in the aging process, they got dephosphorylation. Then dephosphorylated ANG can transfer to the cytosol, then cleave a glutamate tRNA. So the tRNA fragment transfer to the mitochondria, compete with the LARS2 for the amino acylation, especially leucine amino acylation, then affect the mitochondrial translation. So the final result for the mitochondria is the Christie structure got disrupted. Then the biosynthesis enzyme GLS released from the Christie, then glutamate metabolism is down. Then glutamate level is decreased, then affect the learning memory behavior. So we think this study answers a very key question in the brain aging, because during brain aging, learning memory impairment is a key feature. Then this glutamate tRNA fragment accumulation maybe is a very important reason for this learning memory impairment. So finally, I just want to thank the funding support, SFC, MOS, and the cost, and also thank my collaborators from Tsinghua, European USTC, CIPS, and WashU. Okay, thank you, everyone.
Okay, thank you, Dr. Liu. I'm sorry for person got bothered by the black boxes.
That's okay.
But there are,
yeah.
I think it doesn't affect that much. I think it should be fine. Okay, so there are two questions I will ask for the audience. The first is, do you know what happened to ribosomal RNA transcription and RNA POL1 localization inside the nucleolus upon low nucleus non-coding RNA knockout?
Yes, it's actually, we analyze the ribosomal RNA levels. After knockdown or knockout, ribosomal RNA level is changed. But interestingly, we found in different tissue, in different cell types, ribosomal level change differently. In many dividing cells, especially like embryonic stem cells, when we knock down LAUNA, knockout LAUNA, ribosomal level is down. But in non-dividing cell, especially in the neurons, the ribosomal level seems like increase a little bit. So we really don't know what's going on in different cell types. Maybe that's a different mechanism. So hopefully that answers your question. Yeah.
Okay. And the second question is, what potential implications might have for developing therapeutic strategies to mitigate age-related cognitive decline?
Right. I think this question is related to the TSRA, the tRNA-derived fragments. Yeah, it's actually a very important question. So we know in brain aging, especially in the different brain region, accumulated the tRNA fragment is actually a little bit different. We really don't know what's going on, but right now, for this paper, for this study, we only focus on the glutamate tRNA fragment because it's really accumulate only in the neurons, not in other cell types. So yeah, maybe it's a really good target because it can really specifically target this particular tRNA-derived fragment only in the neurons. so they can minimize the side effects. Yeah.
Okay.
Um,
Okay. So
Okay.
next is other tRNAs besides of tRNA glue could be detected during aging.
Yes, we do detect other types of tRNA besides this particular one. But this study will only focus on the glutamate tRNA because the number one accumulation is for a glutamate tRNA fragment. So that's why we only focus on the glutamate. Many other tRNA fragment accumulation, especially in the different brain regions, you really don't know the reason. Yeah, maybe related to different enzyme expression in different cell types. Yeah, something like that. Yeah.
Mm.
Thank
Okay.
you.
Okay, so maybe the last questions due to time limit. Do we know what's driving decreased ANG phosphorylation? Could this be targeted with a small molecule?
Yeah, this is really a good question. We really don't know like how and why NG got dephosphorylated. Yeah, we try to find this phosphatase, but we so far has no luck. I think another way is to find the kinase. Maybe the kinase decrease or phosphatase increase. Either way is possible. So I think either way we can use or design a small molecule to target this. Yeah, it's possible. Yeah.
Okay. So I think the last one is the collaborative ask request question. So I think you can contact Dr. Liu for collaboration. Okay,
Sure.
so sorry, yeah. So
Thank you.
so due to this time limit, so we have to end up the seminar series. I'm very grateful for Dr. Yang and Dr. Liu to present their innovative technology and this very impressive mechanism of work. So thanks for your joining. We will see you next time.
Okay.
Bye.
Thank you. Thanks.
Bye.
Bye.